Hello, everyone. It's time to get ready for another week of Organic Chemistry Lab. So this week we're going to be looking at using a palladium catalyzed reaction, specifically using the iodovanillin we made last week as a reagent. All right. So in experiment 7, we went ahead and performed an iodination of vanillin. So we took our nice, beautiful vanillin molecule and we threw an iodine on it. And at this point, you've probably been wondering for 2 weeks, why on earth did we do that? Well, one of the reasons why is there is a nice set of reactions known as coupling reactions. So this specific one is the Suzuki coupling reaction, where it's able to very targetedly link two different groups. And this one uses a palladium catalyst to do this. And one of the reasons why we love coupling reactions is they're very good at making carbon-carbon bonds. Now, as you're going to see through this semester next, carbon-carbon bonds are actually fairly tricky to make, and they can be fairly tricky to make, especially if we're trying to do so selectively. Now, by using a catalyst, specifically in this case palladium, I can very specifically couple the carbon attached to a boron a boronic acid or a boronic ester or boronic ether to a species containing an iodine or other halides. And when we do this, we are going to make a brand new product via this cross-coupling reaction because I coupled two different species. Now, again, it is worth noting this reaction does occur under this reaction does occur under slightly basic conditions. So thus we're going to need to use the potassium carbonate. One of the things this carbonate will do is since we're working in water, that carbonate will also provide a hydroxide group that can go ahead and attack that boron via an SN2 reaction to result in the release of the boronic of a boronic acid, and again generating our desired product. Now, to perform this reaction, first thing we're going to be doing is mixing all of our reagents together in a 50 milliliter round bottom flask. To this, we're going to go ahead and add a condenser, so very similar to what we did in the original iodination of the villain, but this time we don't need to actually have the addition funnel as well, making it a much simpler setup. Then we're going to reflux this mixture for 30 minutes. And again, remember that timer starts as soon as it starts boiling. Once we've done this, we're going to have to cool back to room temperature. And because the whole reaction occurred under basic conditions, and I need to make sure it's under acidic conditions to generate a nonpolar product, we're going to go ahead and acidify this mixture with 2 molar HCl. And we're going to go ahead and test that it's acidic using litmus paper. Once we have this, we should have a nice solid product dissolved in water, because again, we have a nice non-polar product in water. So what we're going to do is we're going to dissolve that product using ethyl acetate. And then we're going to go ahead and gravity filter because we still are going to have a little bit of that palladium carbon catalyst left behind. And again, note, this is very much in catalytic amounts. So then this should give us a nice mixture of the water, carbonate, boronic acid, and our desired product. So we're going to go ahead and use a separatory funnel to try and extract all of these aqueous components, the water, the carbonate, the boronic acid, and again, now the HCl from now our ethyl acetate and our desired organic product. So again, we're going to use the separatory to funnel to separate this. We're going to go ahead and collect our organic layer. And then what we're going to do is take our aqueous layer and add ethyl acetate 2 more times, each of these times collecting more and more of our product from that aqueous layer. Then what we're going to do is we should now have 3 fractions of our organic layer. So again, 30 milliliters total of our ethyl acetate. We're going to combine those 3 fractions in the separatory funnel, and now we're going to rinse with brine, which is more or less a fancy way of saying salt water. That salt water should go ahead and help remove any of that remaining polar residue. And again, the only thing that should be left after we drain our organic fraction should be, again, that ethyl acetate and our desired product. plus a little bit of trace water because we kept mixing it with water. So we can go ahead and remove that water with the magnesium sulfate. And just like we did in our caffeine extraction, go ahead and remove that mag sulfate using gravity filtration. So then we should be having, the only thing left should be a nice liquid of our desired product and the ethyl acetate. Now we have to get rid of that ethyl acetate. And so we're going to do this using, by putting the sample on the hot plate, warming it to a nice light temperature, and again, using air from your hood to essentially blow off the extra acetate. One way you can think about this is a very rudimentary version of kind of what we're doing with the rotovat. So in order to actually conduct this lab, what do we need to do beforehand? So first thing you need to do is go ahead and review Morg chapter 2 over green chemistries. We're going to be invoking some of those principles. Then don't forget to read the lab manual for experiment 8. And hey, congratulations, you're watching this video. Then don't forget to complete the pre-lab quiz before lab and fill in your pre-lab notebook. Again, with the pre-lab notebook, don't forget to at least have a rudimentary version of a step scheme so you have a rough idea of what we're doing at each step. It can really help to cognitively bring things together. And again, all the relevant molecular weights and densities. That can be really important in this one. And again, answer the questions to consider. Now, once we actually show up in lab, main thing we're going to do is, so first we're going to meet in the chem lounge, the last of our student safety presentations. Then you're going to set up your round bottom flask and your condenser and then reflux that whole mixture for 30 minutes. Then we're going to cool that reaction back down to room temperature. Again, once you're able to more or less palm your round bottom, you should be good to acidify your sample. And again, you're going to add HCl until the sample is now acidic, and you're going to check with litmus paper to verify. Then, with our newly acidified sample, you're going to add about 8 to 10 milliliters of ethyl acetate to dissolve that sample, and then we're going to filter out that solid residue, again, mostly that palladium. Then we're going to add this whole mixture to the separatory funnel. Now, one of the big things you should pay attention to is last time we used dichloromethane as our organic solvent. Now we're using ethyl acetate. So go ahead, quickly verify whether the water or the ethyl acetate is going to be the top layer or the bottom layer. That's going to change how we separate the system. Now, the big thing here is we're going to be extracting the organic layer. Now, last time our organic layer was on the bottom. This time it's going to be on the top. So in order to do this, this means that in order to get to that organic layer, we first are going to need to remove the aqueous layer, and then we can collect that organic layer. And we're going to have to do this 3 times. So we should have 3 organic layers at the end. You can collect them in the same beaker each time, or just like if you're if you're trying to get egg whites, you can collect the organic layer in a separate beaker each time in case you get too much water in it. Then what you're going to do is combine all of your organic fractions back together, add it back into the funnel and add brine. Then you're going to recollect that organic layer after this final separation. Then we need to dry that organic layer, again, using the mag sulfate, and we're going to use gravity filter. So again, no vacuum filtering this week, just 2 different gravity filters. Then your nice dry organic layer that no longer has any water, you're going to go ahead and place it in a dry beaker, put that in a warm hot plate, and then use the air from your hood to go ahead and slowly and very slowly blow on the sample to essentially remove any of that organic solvent. If you have enough time, and only if you have enough time, you can recrystallize your product using warm acetone to dissolve the sample and then warm water to crystallize it back out. Then if you really have, you can take that sample, scrape it into a tarred and labeled vial, and don't forget to save some of that sample for a melting point. So again, loading a melting point too. If it turns out you have a little extra time after this, go ahead and talk to me about running an IR spectra. Now, when you're running the lab, couple big reminders. Pay very careful attention to time because there's a lot of parts going on, and I don't want you to start an optional stage if you don't have enough time. And again, remember, whenever you're heating your sample, you should be running the condenser. Otherwise, you could actually evaporate product and definitely your water right out the top. And again, also make sure that when you're counting your reflux, your 30-minute timer starts when the sample begins to boil. And think carefully, if we're using an aqueous solvent, what should I set my temperatures to on my hot plate? Again, also to save yourself time in class, make sure to pre-calculate the amount of HCl needed to acidify your sample and be very cautious as that potassium carbonate base we're using is diprotic. So you need to make sure you have enough HCl to protonate it twice. Then when you're using the separatory funnel, remember if I need to remove that top layer, first we need to go ahead and pour the bottom layer right off the bottom, collect that, and then we can collect the organic layer before adding that aqueous layer back to the funnel. So again, there's a lot of extra steps compared to last time. Now, this is always a question when it says to rinse your solution with brine. Brine is just a fancy old-fashioned word for an aqueous salt solution. So again, which brine we use will often switch, but this is usually going to be a nice sodium chloride solution. We're going to make sure to add enough magnesium sulfate to the organic layer until the magnesium layer sulfate stops clumping. That's usually a sign that we've taken up all of the water. At this point, you can go ahead and filter. And then again, to use the air to evaporate our solvent off, we're going to use the air line in the hood. So again, you have a valve for air, and you're going to use that to evaporate that ethyl acetate, but make sure to turn that pressure on low. If you turn it on too high, you can quickly splash sample out of your beaker, causing you to lose sample. And again, only recrystallize your product if you have at least a half an hour left in class, and that probably would still be cutting it a little bit close. If you don't have that much time, go ahead, skip the recrystallization. Your product can be a little less pure. It's fine. And again, similarly, only perform the IR if you have time and work with the instructor or TA to show you how to do so. Now, once you're all done, as usual, we're going to need to load our post-lab notebook scans. You're going to need to write up your findings from Experiment 7 and Experiment 8 in our final big project of the semester, a full lab report describing what you did over these two labs. That report's going to include an introductions, so essentially summarizing what we did and kind of why, a methods detailing how we did it, a results section summarizing what you found, and a discussion section telling you what it means. And again, instead of trying to treat this as separate labs, this is a single project done over several weeks. That does not mean I'm expecting a week 1, week 2. Instead, you can go ahead and just put it over each individual step. And again, for the results, we're going to need to include information from both parts. So for both parts, you're going to need your percent yields. Again, both on each step of the synthesis and each step of the purification. So what was your percent yield before crystallization, after crystallization, and so on. Also, make sure to include information from the IR and NMR spectra from each time, how you can go ahead and distinguish the samples. If you do not get to run your own IR spectra, go ahead and treat the ones provided online as a version of yours. And again, make sure you include your melting point data at each step. And again, summarizing, most of this information should end up going in tables with potential IR and NMR data in figures. Now for discussion, big things you should be talking about is what is your yield? How could it have been better? What could you have done differently? Was it good? Was it bad? And so forth. What was your purity of your sample? So if you don't have any IR, this is mostly going to rely on your melting point data. And again, overall evaluation of how green this reaction is. So think very carefully about how much waste you've been generating each time and what you could do better next time. All right, a rough draft of this report is due on Canvas on the 25th of November. And then I want you to take a little bit of time over that Thanksgiving week to go ahead and do a quick peer review of someone else's paper. That peer review is going to be due on the 2nd of December. And then taking that information that someone else provided you, a final draft of the report is going to be due on December 12th at 4 p.m., at which point I'm going to have just enough time to grade them all before the end of the semester. All right, so so now we're really coming to the end of semester, and those are the major things to be looking forward for this week in lab and what to anticipate afterwards. Look forward to seeing you then, and take care.